Okay, let's go ahead and get started. So we left off yesterday talking about translation. So I'm going to go ahead and we'll finish with translation and move on to biotechnology today. Go ahead and share the screen with you. Maybe. Let's see. Okay. So the eukaryotes have this whole nucleus thing that they have to deal with. They have to get the transcripts out of the nucleus and to the ribosomes. And so in eukaryotic cells, there are a couple of different places that we can find ribosomes. We can find them free floating in the cytoplasm. And this is great for any proteins that need to be in the cytoplasm, proteins that don't need to be shipped around the cell. For proteins that might need to be shipped around the cell, the ribosome is going to be found in the endoplasmic reticulum or attached to the endoplasmic reticulum. So the rough ER is going to be the part of the endoplasmic reticulum that's studded with different ribosomes. And the purpose of having them there is because the endoplasmic reticulum is basically the beginning of the shipping chain within the cell. So if we need to ship proteins to either other organelles in the cell, to the membrane, or to the extracellular environment, we have to have them start in the ER so that they can be packaged and shipped to other places. So here's a little video about protein synthesis. Again, if you don't like this animation, you can find lots of animations on YouTube about how these processes work.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Okay, again, we will talk more about the protein processing and protein shipping as we talk more about functions within the cell. But that idea of transcription, translation, and protein formation is what I'm hoping you'll get out of these 2 sections. So the last thing that we'll talk about in this section on translation is what happens when you have a change in the DNA sequence when we consider, the effect on the potential protein product. So we know that the gene sequence is linked to the protein sequence, and we know that protein sequence is linked to protein function. So if we look here at our gene sequence, remember we know our first amino acid is going to come in here and be a methionine, and then we can read the chart. Again, don't expect you to remember any of these others. You are able to read the chart and make your proteins that way. So this is our primary sequence: methionine, valine, glycine, serine, leucine, threonine. There's our protein. It will form these hydrogen bonds between amino and carboxy termini, either a beta sheet or an alpha helix. And eventually that secondary structure of alpha helix, beta sheet will fold in on itself even more, and we may get things like ionic bonds between the R groups. We may get disulfide bridges. We may get additional hydrogen bonding between some of the different R groups. And if we have multiple subunits that make up a protein, we can eventually get this quaternary structure. But depending on the protein, if it's a single subunit protein, that tertiary structure will give us the function. If it's the quaternary, then we'll get the function once we put all of the subunits together. So when we have a change to the DNA sequence, does that change the protein sequence, and does that change the protein function? So anytime we have a change to a DNA sequence, we call that a mutation. And that is a change that is heritable, meaning the cells that are the descendant of that cell will also have the mutation. They'll also have that change in the genetic material. So it doesn't necessarily mean that it's going to necessarily be passed like to your offspring. The only time a mutation would be passed to the offspring is if it were in one of the germ cells, in the sperm or the egg. But you can acquire mutations to your skin cells if you go outside and get a sunburn, and then all of the descendants of those skin cells will still have that genetic change. So that's what we mean when we say heritable. So in a single-celled organism, it obviously would be passed to the next generation. So we have a bacterium, the bacterium replicates, one of the bacterial offspring acquires a mutation, all of its offspring will have the mutation. But when we're talking about a multicellular organism like us, the only type of mutation that would be passed on to the next generation is going to be a germline mutation. Again, a mutation that occurs in a sperm or an egg. A somatic mutation, somatic means body cell, would not be passed on. So again, if somebody's a heavy smoker and they get mutations in their lung cells because of smoking, that is not passed on to their children. Skin cells, not passed on to the children. Only germline mutations would be passed to offspring. And there are lots of different kinds of mutation. Some of the more dramatic types of mutation can include changes in chromosome number. We call that aneuploidy. We'll talk more about chromosomes and how chromosomes are inherited a little bit later in the term, but just very briefly, human cells, with the exception of gametes, human cells should contain 46 chromosomes. We have 23 different pairs, and those pairs are numbered 1 through 22. And then the final pair, that 23rd pair, is the sex chromosomes. So you can have individuals with 2 X's or individuals with an X and a Y. And that is the standard number of chromosomes. When the number of chromosomes changes, there are dramatic consequences on the offspring. So what they're showing you in this image is an individual who has Down syndrome. Down syndrome is caused by what we call trisomy 21. So instead of 2 copies of chromosome 21, this individual has 3. There are only 3 trisomies that are tolerated in humans in these chromosomes that are called autosomes. They're not the sex chromosomes. Trisomy 21, trisomy 18, and trisomy 15. Trisomy 21 is the most mild. Trisomy 15 and 18 are very, very severe. In the sex chromosomes, we can tolerate more. You can have individuals who are born with only a single X and no other chromosomes. You can have individuals born with 3 X's, 4 X's, an X and 2 Y's, 2 X's and a Y, an X and 3 Y's. So with the sex chromosomes, we play around a little bit, but with the other chromosomes, we call them autosomes, that is a very significant mutation. So this is, again, a severe mutation in a change in chromosome number. We can also have what we call large scale changes in chromosomes. So pieces of chromosomes that go missing, or pieces of chromosomes that get flipped around or get moved from one to another chromosome. This generally happens when we have errors in meiosis. And we'll show, when we talk about meiosis, we'll see how that can happen. Again, we don't tend to tolerate these types of issues very well. There is one that I'm thinking of off the top of my head that's a deletion of part of chromosome 5. And the condition in English, it's called cry of the cat. I don't speak French very well, but the real name is in French, so that's why I'm not even going to try it. In English, it's cry of the cat, because the children have significant mental defects, and they cry a lot, and when they cry, it sounds like they're meowing like a cat. So very significant consequences when we alter chromosome structure and number. More commonly, and something that all of us will experience at some point in our lifetime, are what we call point mutations. Now, point mutations are going to be small changes to the DNA, like changing a single base. So these are much more frequent. They usually have far fewer consequences, and they occur all of the time in our cells. So how do these things happen? How do we get, let's say, just a point mutation? We can get a point mutation spontaneously. And this is simply because our replication system isn't perfect. We talked about the idea that the majority of the time, our DNA polymerase can do proofreading. It can realize it made a mistake. And even if the DNA polymerase didn't catch the mistake, our mismatch repair system should catch the mistake. and fix it. But the error rate, so the errors that don't get caught, the rate is considered to be approximately 1 in 1 billion base pairs every time the cell replicates. And we have 3 billion base pairs in our cells. So spontaneously, every time one of our cells replicates, it should acquire approximately 3 point mutations. So after generation 1, we have 3 point mutations. Generation 2, we have 3 more. Now we have 6. Generation 3, 3 more. Now we have 9. And so what we see over the lifetime of an individual is the increasing number of point mutations and the increasing possibility that they could begin to affect a gene that's important. So we have lots of regions of our DNA that are not necessary for making proteins. If you get mutations in there, usually not a problem. But if you start to get mutations inside of protein coding regions or inside of a promoter necessary to begin transcription, we can start to have issues. And so that's why things like cancer, which are caused eventually by mutations, are far more common in elderly patients rather than young patients. I said far more common, but of course we all know it is possible for young people to get cancer too. There are also normal, though rare, chemical changes to DNA. So sometimes DNA can be spontaneously methylated or have other types of chemical groups added, which can be a mutation as well, but it's fairly rare. Far more commonly, we think of induced damage to DNA. So if we have a failure to repair, if we have damage to DNA and failure to repair, we get a much higher rate of mutation than with spontaneous mutation. So things that can damage DNA include X-rays, UV light, and chemical mutagens. So anything that causes a change to DNA is called a mutagen. And I always laugh because I grew up in California. I moved to Texas to do my PhD and postdoc back in 2007. And then I moved back here in 2015. And I don't know what happened, but in the time like between when I left and when I came back, there was this proposition passed called Prop 65. And now every time I go into like any building, it's like, Prop 65, this building contains chemicals known to cause cancer. Like I walk into Starbucks and I'm like, well, whatever, it's totally worth it. Everything is a chemical that could potentially cause changes to your DNA, so therefore it is a potential carcinogen. So that's what that means. When you see that sign, it's basically telling you there are chemicals in here that we know can change DNA. It doesn't mean they will, it doesn't mean you'll be affected by it, but we know it's a possibility. And so we're just letting you know. And so when I came back, I moved back here, I was like, where did all these signs come from? And my mom said, well, you know, it is what it is. So everything is gonna try to change your DNA and can potentially cause cancer. The problem is these types of things like radiation or these chemicals cause so much damage to your DNA that your repair system can't possibly catch everything. And so that's why you see individuals who are outside a lot, landscaping, agriculture, or over over tanning are far more prone to develop skin cancer because they're constantly bombarding their skin cells with UV light, causing damage that their body just can't repair. Okay, so when we have point mutations, we can also classify them by the exact type of small change to the DNA. Most commonly, we would see what we call base substitutions. So the total number of bases stays the same, and only a single base pair is altered. So this would be, I changed a G to an A C to a T, whatever. And so we changed a single nucleotide and swapped it out for a different one. We can also have what we call insertions and deletions. sometimes people will classify these. They'll use the term indel, I-N-D-E-L, indel, to refer to either an insertion or a deletion. So this actually leads to a change in the number of bases, either an insertion of bases, one or more, or a deletion of bases, one or more. And when we talk a little bit more about this, you'll see I usually argue that indels, insertions and deletions, are generally far more dramatic on the protein product when compared to base substitutions, and we'll see why. So here's an example gene. That's what the gene looks like. You have your two strands. They are complementary. Everything is good. The mRNA would look like this. So, we would transcribe from this bottom strand. We generate our mRNA. Here's going to be our stop codon. Here's our start. Everything is great. Our protein product is perfect. Look how happy we are. Now let's start playing with that sequence. So here's our sequence. If we go back to the original, we've changed a CG pair that's 4 in from the left to a TA pair. So we went from CG to TA. So how does that affect? Well, it changes our codon. Instead of having a C, it now has a U. All right. What effect does that have? Oh, actually, because of, remember, this concept of degeneracy of the amino acid code, this codon, GGU, gives us the same amino acid as GGC. So, no big deal. So we call this a silent mutation because it has no effect on the protein product. So this is basically, it's a neutral, harmless mutation. Not beneficial, but not harmful. It happened, but everything is still the same, so we're good. We then can have what we call a missense mutation. So in this case, we've changed, this was a GC pair, and we've changed it to an AT pair. So let's see what happens. We've now changed our codon. If we remember, this codon was GGC. That gave us glycine. So let's see what happens here. Now we've gone from glycine, which was the original, to serine, which is not the intended amino acid. We call this a missense mutation because we basically changed the word. It should have been glycine and now it's serine. It's missense. Will this have a dramatic effect on the protein? It's hard to say. It really depends. In some proteins, there are very particular amino acids that are absolutely critical. And if you change one of those, yes, you do change the entire function of the protein. But in most proteins, there are also lots of amino acids that even if you change them, it doesn't make that much of a difference. So in this case, it's really hard to predict. We would have to know, what does the normal protein do? And is that glycine actually important for the normal function of the cell? But either way, regardless of whether or not this is harmful to the protein, not harmful to the protein, or beneficial to the protein, we call this a missense mutation because we have swapped out an amino acid. The final base substitution result is going to be what we call a nonsense mutation. So in this case, we have swapped the fourth nucleotide from an AT pair. So it was AT, now it's TA. So that is going to change our transcript. So now our transcript, instead of reading AAG, it reads UAG. And that's a stop codon. So now our protein is simply methionine, when it should have been methionine, lysine, phenylalanine, and glycine. This is generally an issue. I mean, if it was like the second to last codon, yeah, probably not. But what is the chance that that's going to be only the second to last codon? It's far more likely that something like this is going to happen somewhere in the middle of the protein, and you lose a lot of the structure, and of course, you therefore will lose a lot of the function. So nonsense mutations are, I would say 99% of the time, going to be very harmful for the protein. The reason we call it nonsense is because you get no amino acid. So there's nothing, no sense, so nonsense. So to kind of summarize this section, we can have silent mutations that give us the exact same amino acid. We can have missense mutations that give us a different amino acid. We don't know whether or not that will be harmful for the protein or not, but we do know it's a different amino acid. And then we have nonsense mutations where we now have a premature stop codon, and we truncate or shorten the original length of the protein. So that's what can happen with a single nucleic acid change, swapping out one base for the other. Now we'll see what happens if we start removing or adding bases. We do indels. So what they've done here is we've taken our original gene sequence and we deleted this single pair, this TA pair that was right here. So now we end up with, this is what we call a frame shift mutation. So what happens is, right, we read in groups of 3. So we would be reading AUG, AAG, and it should be another T here. So we should be reading UUU, GGC, UAA, and that should be our protein. But because we've removed a nucleotide, so we've removed a nucleotide from the mRNA, we basically took out the ability to read it appropriately. We've shifted everything over. So we took out the U that was supposed to be here, and we shifted what we call the reading frame, that group of 3, over. Now our protein is methionine, lysine, leucine, alanine, on and on and on. We don't know where the stop is. We've shifted everything over and we no longer have a stop codon. So insertions and deletions, when they lead to this type of frame shift, are going to dramatically alter protein function, because they are going to dramatically alter the amino acid sequence. If you add or delete 1 or 2 nucleotides, you are going to change everything downstream. If you add or delete 3 nucleotides, you're either going to remove a single amino acid or add a single amino acid, and everything else downstream should be okay. Who knows what change that single amino acid would have, but 1 or 2 nucleotides, I mean, you've changed everything about this protein. So the take home for the rest of translation, tRNAs recognize codons in the mRNA because the tRNA has a section called an anticodon that will bind to the codon. tRNAs bring in amino acids to help grow the polypeptide strand. Ribosomes are a complex made of proteins and a ribosomal RNA, and they help to guide the mRNA and move it so that we can bring in the tRNAs and build the proteins. During the step of initiation, the mRNA will encounter the small subunit of the ribosome that will recruit a tRNA carrying methionine, which will recruit the large subunit of the ribosome so that we can begin protein synthesis. During elongation, tRNAs carrying amino acids will come into the A site of the ribosome. The polypeptide chain that's on the P site will be added to the A site. Everything will shift over, and the uncharged or empty tRNA will exit from the E site, and a new tRNA will come into the A site. And so we read each codon as a group of 3. During termination, one of those stop codons, one of those 3 stop codons enters the A site, and that does not recruit tRNA. It recruits a protein called a release factor. That release factor will basically clip the polypeptide from the tRNA, allowing the new protein to be free. If we're in eukaryotes, that protein may go into the rough ER. The protein may stay in the cytoplasm. In prokaryotes, it's going to be in the cytoplasm. And then the protein can be folded into its secondary and tertiary structure. In eukaryotes, initiation begins in free ribosomes, but if the protein needs to be shipped, it can be attached to the rough ER. Mutations in DNA sequence can lead to changes in the protein. Those may or may not be harmful. They may or may not be dramatic changes, but that's what we can see. Base substitutions could lead to silent mutations in the protein where we have the same amino acid. Missense mutations in the protein where we've swapped out the amino acid, or nonsense mutations in the protein where we have ended protein synthesis. We have incorporated a stop codon. Insertions and deletions change the reading frame of a gene, and we now go from a normal protein sequence to something that is completely different from what should have been. So insertions and deletions are generally far more dramatic than a base substitution. Okay, any questions about translation before I break you into your groups to answer a question?
For the polyribosomes, is that only in prokaryotes, or is it?
Yes, only in prokaryotes.
Okay, and then also for the nonsense mutations,
Yes.
does that affect all the way down to the end of the DNA, or just that gene?
It would only affect that gene, because every other gene is still going to have its own promoter. And so even if, you know, the little change in the chromosome occurs of that single nucleotide, the promoter for other genes is still going to be intact, and it'll still be recognized, and their mRNA will still be the same. So it's not necessarily the fact that there's a change to the DNA, it's what happened when that DNA was transcribed and gave you that different mRNA. So the mRNA of other genes should be totally unaffected by a single mutation in a different gene, because they'll still do transcription the same. It's translation that becomes the issue for that single mRNA fragment. Okay. Any other questions? Okay, so let me go ahead. Oops, that's not what I want to do. And put you in your rooms. I have opened up, that's not the question I wanted. Hold on. Let me open up the question real quick, because I know that I didn't do it last time. Okay, the question should be available, and I've created your rooms. I'll bring you back in maybe 5 minutes. Shouldn't take too long.